Okay, good morning, everybody. If you've got any questions while I'm moving through here, just stop me and I will try to deal with that. I'm not seeing the chat stuff right now. All of that. Oh, there it is. All right. So we're going to start this morning with chapter 13, Control of Microbial Growth. And part of me feels like this maybe should have been the very first chapter that we think about as we start with microbiology, trying to think about all sorts of different potentials for how we may control microbes. And there's some details about lab and uh, things like that that could potentially be the starting point. And as I start this chapter every time and see this image, it makes me think of your lab, like the very first one when you do some environmental swabbing, although you didn't go outside, but you swabbed all sorts of different things just to see some potentials. And looking at this car makes me stop and wonder, uh, how clean is your car? How clean is clean? Might be a good question. There are numerous things that we come in contact with all of the time, driving in our car, touching different components, and we see all of these different areas, touch points, uh, where they did some swabbing and basically trying to see what, not really what, but how many things are potentially growing there. So they're looking here at the average number of CFUs. That stands for colony forming units. You guys have seen colonies in the lab as you look at your petri dishes and streak for isolation, and seeing these individual colonies is really what we're trying to go for. So we're not talking about individual types of bacteria that we've found here, although I'm sure they could have done further studies to figure out those details. Uh, just make sure. Um, that's not what we're looking at here. So you can see as we move through here, there are definitely things that are more common to be touch points, like the door latch or the center console, the gear shift, uh, maybe the radio knobs, right? And then some other stuff like the window control over here. Maybe not used so much. Maybe the window's up because we're always using air conditioning. I don't know the answer to that. So as we get down in here to the console, there's other potentials like maybe spills and things like that that potentially lend to some greater opportunities for some microbes to potentially thrive. So you can see there are numerous instances where there are a large amount of colony forming units. And I would assume in these circumstances, probably a lot of variation. When you did your environmental swabs, my guess is uh, there were numerous types of colonies that you saw on the plates. A lot of variation, a lot of diversity. Maybe you found some fungus on there, uh, all sorts of opportunities. So we think about things like, I mentioned as we started this, how clean is clean? Uh, we, clean our car, maybe we do, vacuum it, maybe clean some of the interior, but how often does that occur? You might think about your house and, cleaning within there. And it makes me think about things like, 5-second rule. I'm sure you've all heard of that. Maybe you practice it. Something drops on the floor, a food item, and maybe you quickly pick it up and decide it's still able to be consumed. Others may think, nope, as soon as it touches the floor, that's garbage. Truly kind of depends on the circumstances. Might depend on the floor. Uh, so, uh, those are things that, makes me stop and wonder just the littlest bit. What sorts of potentials are we coming in contact with all of the time? Uh, so when we're thinking about microbiology, there are all sorts of different levels. And we've already described some of this stuff previously. We've talked a little bit, but we're really going to define some of it in the next couple of slides. So, the top level of concern is the BSL-4 biosafety level 4 types of laboratories. We've mentioned some of this previously, the spacesuit sort of situation where air is piped into that, blowing that suit up. You can see gloves and tape here, fume hoods that have filtration apparatuses, probably some UV sterilizing lights as well. We'll hit all of these things as we move through here. Uh, and the labs have all sorts of other setups with uh, positive pressure pulling air in from out. None of the air in this lab goes out of the lab. It moves through this hood, through filtration devices, keeping everything within. Uh, so we definitely don't think about those situations here. And typically, we don't think about moving past this BSL level 2. Uh, and none of our labs are equipped to move up above that. So this figure shows the biosafety levels, and this is from the CDC, but this is something that is almost universal. May call it slightly different things, but it's something that we recognize not just in the US as far as uh, recognizing potential levels of concern. So it truly depends on the circumstances. It depends on what it is we are working with. The type of lab is set up to deal with those respective things. And potentially without that, you're barred from working with any of that. So almost everything that we work with in the lab is at this bottom category, a biosafety level 1. And in these situations, the microbes are, I would like to say, safe. They're not known to cause disease in healthy individuals. They pose very low or minimal risk to workers or the environment. There are definitely potentials where some of these things may cause illness in an individual that's not classified as healthy. But typically, we don't think of these things as really being true pathogens. Organisms that are oftentimes classified as endemic, so something you just expect to see there. Um, Not something posing serious threats. So one example is some E. coli. There are boatloads of different types of strains of E. coli, but some of them we classify as non-pathogenic. Maybe like the E. coli that resides within our gut, uh, not something that we think of as truly causing serious concern. Not that you would want to ingest it. Stepping it up a notch, we see Staphylococcus aureus. This is an organism that we also work with in the lab, but it's a level up. So these also something that we would think about being relatively common, and ultimately these bacteria are found on our bodies in various areas. I wouldn't think of them covering my entire surface of my skin, but there's definitely potentials to find some Staph aureus. So There are potentials for some disease situations. Staph can definitely cause infection. And so a little bit more precautions are warranted in these situations. And when we think about stuff like this, our laboratories are set up to handle these situations. We see in the lab sinks with soap. We see the biohazard buckets where all of our petri dishes go. There are tubs for the test tubes, all of that stuff. The petri dishes are going into the bucket and they get dealt with separately, but the test tubes, all of that material goes through an autoclave, gets disinfected, and then cleaned. Uh, so there are also potentials where we think about that in level one. We want to do some decontamination, but you need these opportunities. We don't see each of our individual labs uh, with autoclaves, but in the stock room down in the basement of the biology building, there are autoclaves that deal with all of this stuff. So the measures are in place to ensure that we can deal with this. Stepping it up a notch, and like I said a second ago, we only go to BSL level 2 here at ISU. Other institutions have the potential to bump it up to BSL level 3. When we get to BSL level 4, we're thinking about government labs, CDC and NIH, that have the capabilities to work with these types of microbes. So the level 3, these are organisms that definitely pose more of a threat, serious or lethal diseases. And we think about more stuff like aerosolized uh, potentials for infection. So respiratory transmission, definitely issues. These BSL level 3 labs don't require the same spacesuit like we see here, but they definitely have other circumstances like the fume hood, as most likely everything being worked with in there. Individuals working in these labs most most often are wearing respirators to ensure that aerosol isn't a potential uh, for them. Stuff like tuberculosis is an example with that. The text goes through and mentions a handful of others. There's some different viruses. HIV fits into this BSL level 3 category. Bumping it up to the top, we think about things like Ebola and Marburg viruses. These are both hemorrhagic viruses. They pose serious threats and are often fatal. So we can see this pyramid with the level of risk, and these ultimately things that I would recommend you try to avoid. Uh, it always, as I think about these spacesuit situations as you leave the house to go to work, that truly could be the last day you leave, right? You hope you hope everything is good. So we think about all sorts of different ways that we control growth. And really, in this chapter, we're thinking about physical mechanisms to deal with microbes. When we bump over to the next chapter, we're going to think about more like our bodies and antibiotics and how we can use different substances to target respective microbes. Uh, but a lot of this we can think about, like I was saying, laboratory situations where we're trying to ensure that what we're working with is what we're truly putting on the plate and not something that we've picked up from the bench or potentially from our own bodies. So we go through numerous protocols to ensure that our situation, our lab area, is aseptic. So when we think about these mechanisms, there are different categories, depends on what we're truly thinking about. So the first bit for use on fomites, fomites are basically all sorts of inanimate objects that are, we come in contact with. The car that we started with, all of those different touch points, your cell phone, whatever it may be, some inanimate object that gets touched, and then potentially leaving microbes on it. Somebody else touches it, and then maybe they get ill. So, dependent upon the type of fomite, we've got different opportunities to try to deal with cleansing them with maybe the hope for sterilization. So not everything can be sterilized. But with that, and I'm going to start at sterilization and then we'll work backward. We think about the complete removal of organisms, killing all vegetative cells, killing those organisms that have the potential to form endospores and killing those spores as well, and also viruses. So getting rid of it all right? And oftentimes when we think about these situations, the first thing that really pops into my mind uh, is an autoclave. So pressurized steam, high heat, and pressure to get the temperature up enough to actually kill those spores. But in circumstances where autoclaves aren't able to be used, potentially the temperature too great for that substance, different chemicals or radiation may be used to achieve sterilization. The petri dishes that we have all of our media in come in a sleeve, a plastic sleeve. There's 20 or 25 petri dishes in that sleeve. Those plastic dishes can't go through the autoclave, they would melt. So they use chemicals, uh, I believe ethylene oxide, a gas that goes into the sleeve before it's sealed up, and all of the plastic within there ultimately is sterilized. Once it's opened up and you're, dealing with the plates, there you want to work aseptically to ensure that you don't end up contaminating them. So, stepping backward from that, uh, to sanitization and disinfection in these situations, uh, which are probably more common practices, because we're not autoclaving things at our house, but we are trying to clean. We think about sanitizing or disinfecting. So you're bringing down the amount of microbes, but you're not truly not eliminating them all or killing them all, but you're bringing them down to safer levels and making it clean, uh, however you would like to define that. So when we think about sanitizing, we reduce the load, bringing it to levels that public health levels that we would recognize as being safe. Uh, and it kind of depends on the circumstances. Uh, Dishwashing, commercial dishwashing, we think about with different sanitizing agents. There are potentials, as we see over here, the finish that may go into your dishwasher, but oftentimes in a restaurant situation, there's a little bit more uh, of action on that to ensure that those products, the silverware or the dishes, uh, are uh, a little cleaner, if you will. So lots of detergents with phosphates, industrial strength cleaners that have some ammonium compounds within them, work as a sanitizing agent. Disinfection, this is stuff that we think about like uh, cleaning the laboratory benches. We disinfect the benches. And the cytocon is a disinfectant. It works through evaporation as it gets on there and then slowly evaporates away. After it has evaporated, we would say the bench has been disinfected and the potential for microbes has been dramatically reduced. All right. Uh, so heat and chemicals. So as I said, the lab bench, clinical surfaces, bathrooms, chlorine, bleach, and phenols are agents that are typically utilized. Lysol is a disinfectant, and glutaraldehyde. All of these are agents that could be applied, and then given a small amount of time, we would say it's been disinfected. So definitely some differences here between sterilization and disinfection and sanitization. Um, and it truly depends on the circumstances. You would hope if you were in the hospital and you needed some sort of surgical procedure, that the scalpel or the needles or whatever it may be has been autoclaved to ensure that it is truly sterile, right? And not just using a disinfectant. Shifting from fomites to ourselves, this is a different situation. You wouldn't want to bathe yourself in the cytocon that we disinfect the benches with. Uh, Probably not a good idea. When we think about living tissue, there's two different ways that we deal with this, antisepsis and degerming. And in both circumstances, where it's like our sanitization. We're trying to reduce down that load, but your tissues, you don't want to cause damage. So we have definitely different substances that we would employ uh, in these circumstances compared to up above. So antisepsis brings that load down on skin or tissue, and we think about some sort of chemical agent, alcohols, boric acid, peroxide, and iodine are substances utilized for that. And we think about cleansing the skin or cleaning it before surgery, oftentimes betadine applied to that, and then allowed to uh, sit on there for a few minutes before the procedure occurs. Degerming, we think about hand washing. So bringing it down with gentle to potentially firm scrubbing, uh, depending on the situation. Soaps and swabs fit into those categories. And we will go through and talk about some details with this as we progress along with some of these different agents and show some features. So with that, there when we think about the killing of these microbes, we talked before about growth curves and we saw exponential growth, and we saw as those bacteria were growing, they would start to plateau, reaching a stationary phase. And then we saw the decline or the death phase of those organisms. And those situations were basically thinking just, what's up with this bacteria growing in a broth? uh, and you could see them dropping off. This is a similar idea, but in this circumstance, we're thinking about some sort of agent that we might be using. How quickly is it, is it able to, kill these microbes, bringing those levels down to safe levels or to zero potentially. So we can see in this figure, we've got the logarithmic scale in red, and oftentimes this is how we would like to be presenting it because this gives us an opportunity to actually extrapolate out some of the details. With the arithmetic scale, we see more of what I said a second ago, this exponential, but this would be exponential death. So a rapid decline and then kind of plateauing out. But in these circumstances where it looks like we have approached zero, have we actually? So that's kind of the question. And this on the axis showing us our log and then other arithmetic scale as we move down here. So all of those different ways that we talk about dealing with those microbes, whether it be fomites or our own tissues, there are definitely differences as far as how those substances may be acting upon them. And we can see over here the time and the number of cells as we move through here before it's applied. You've got, a billion, and then we start dropping it down every 5 minutes. We're looking at it until after almost an hour, we get down to 10 to the zero. All right. Uh, so we look at this D value as we move through here, one little square within this. This is the time it takes to kill 90% of the population, a 1 log decrease. So going from here to there, right, and from here to there. And that's what we're looking at here, 5 minute intervals as we're moving down. So how long does it take? And it, like I said, depends on the respective substance, its mode of action that allows for that. And like I said, with the cytokine, your lab disinfectant, technically that takes about 10 minutes. I assume when you go into lab, you spray your disinfectant, you wipe it with paper towel, and you wipe it till it's about dry. Uh, is that the best? Maybe not, but the hope is when you're done with lab, you spray stuff on there and you wipe it a little bit, but there's more disinfectant left and it evaporates a little more. And then the next group comes in and they go through the same practice. So the hope is when you show up, your bench has already been disinfected from the previous lab, and you're just kind of cleaning it off a little bit more. All right. Um, so that seems to be pretty effective. All right. As we look at this image, we're thinking about sterilizing, and in circumstances like with our inoculating loop, oftentimes we use a Bunsen burner. Fire uh, gives us the opportunity to heat this inoculating loop till it's cherry red as we look in here, and any organism that could have been on there has been incinerated, and after it has cooled, uh, we would recognize it as being sterile and ready to make those transfers, whatever it is you're doing. As soon as you've done that, you'd pass it back into the flame and re-sterilize it, cleaning it off, and ready to go. All right. This is a bacterio incinerator. This is basically this little incinerator, and we showed this a few weeks back where they were putting a slide in front of it. The inside of this chamber, it's a ceramic chamber. The loop would go into that. It's got some protective screen around it, and it's kind of like a toaster. When this gets turned on, there are heating, come back to my pointer, there are heating coils that go around this ceramic cylinder, and it gets it super hot. So you've got like a little oven inside of here. The loop goes in, and this is important when we think about working with substances that may be more of a potential for an aerosolizing agent. You would want to ensure that everything is trapped within this chamber and doesn't come out. This out on the bench top, but oftentimes these may be found like in the fume hood where you'd be doing your transfers if you were truly working with potential aerosolizing agents. It wouldn't just be out on a bench top like this. Uh, and I like this image with all of the clutter around here. This is like this is a working lab. You see all sorts of stuff in there. So the autoclave, there are all sorts of variations with this, from small benchtop autoclaves to some very large autoclaves that could actually be about the size of an entire room. Oftentimes, a hospital has an autoclave with uh, one side of the room being the dirty room, where everything that needs to be disinfected, it needs to be sterilized. All of those samples or specimens go into that side of the room, and it moves through the autoclave, goes into the door, which I'm imagining is probably what I'm seeing right here. Uh, you would wheel the cart in with all of the stuff. It would get autoclaved, and on the other side is another room that would be the clean room. Once that autoclave run is complete, you open the door on the clean side and pull your material through. That way, materials that have been sterilized don't come back into the same room where dirty stuff is. That's another level of safety to ensure that we're not potentially contaminating. Most of the time, it just comes right back out of there and it's in something that keeps it closed up and remains sterile. So it uses heat and steam to pressurize, and you can make all sorts of adjustments as far as the timing of that. How long does it truly need to be cooked uh, to deal with that? And this uh, very similar, although bigger, very similar to like a pressure cooker for home canning, or maybe an instant pot, if any of you uh, use any of that for some cooking. It's a similar idea, using heat and pressure to do that. And if you do anything like that with an instant pot, you potentially recognize you can cook things at different times. Some things less, some things require more. Uh, and it's the same sort of an idea when we're thinking about this stuff. Oftentimes, like your materials that you get in the lab to inoculate, like those broths or slants, those test tubes full of media, they get autoclaved. The media gets autoclaved in those tubes, and typically those autoclave runs are 15 minutes at 121 degrees Celsius. And when it comes out, we would say that it's sterile. When those substances, whatever they were, have been inoculated with bacteria, we've grown them all up, we see the yellow ferment tubes or growth on those slants, whatever it is, obviously bacteria on it. They go back to the stockroom, they go into the autoclave, but this time it's a different cycle, and we call it a kill cycle or a kill run. It's still 121 degrees Celsius, but the time is increased from 15 to 25 minutes to give it a little extra time to just ensure that it's truly done its job. 15 minutes could potentially be successful, but giving it, almost doubling it up, gives it a little more time, a little more reassurance that if there were spore forming organisms in there, we actually did our job. There are other issues where maybe you cram way too many things in there and you don't properly heat through the entire mass of stuff you put in there, and some things may not be as sterile as you truly think they are. So one way to recognize that is through, as we see these tubes over here, this tape. This is autoclave tape, and we can see one tube. You can maybe see, I'm putting my arrow by it, this white strip here. The tape, like masking tape, has these markers in here, and they're temperature sensitive markers. They turn black, as we see over here, if we've made it to the proper temperature. They only turn black if it's made it to 121 degrees Celsius. If we haven't hit that temperature, it remains colorless. So this is an indicator that your sample has reached the proper temperature. There are some other opportunities where a small vial is put in there with all of the autoclave run. That vial has a spore of Clostridium in the vial, probably more than one spore. It's got Clostridium spores in it, and it's inside. It's in a little vial. This ampule has the bacteria, and it also has broth. And when the sample has been autoclaved, you give it a little squeeze. It breaks the inner bit of uh, of glass that has the spore in it, and it basically inoculates it into the broth. If that changes colors, it indicates that the spore is still viable, and then you know whatever you autoclaved has not actually been effective. That requires a little bit of time, uh, incubation for that to occur, but it's an extra line of reassurance, I guess I would say. Oh, autoclaves. So when we think about this stuff, there are differences when we talk about killing. So a thermal death point, when we think about that, it's the lowest temperature that can be used where we kill everything within there in a 10-minute time frame, right? So what is that temperature? It may require boiling. It may require that 121 degrees autoclave temperature, or maybe even a lower temperature. Lots of microbes that live at our body temperature may not need a super high temperature to kill them. Bringing it up to maybe a steam, uh, you know, 70, 80 degrees Celsius uh, could be sufficient, right? The thermal death time is, and now with the death point, we've got a specific 10-minute time. The thermal death time, now we're trying to figure out the length of time, all right? So how much time is needed to kill all of the organisms at a respective temperature? So now we're keeping the temperature constant. How long? How long would I need to keep something at, you know, 70 degrees Celsius or at 121 degrees Celsius to ensure that it's truly effective? I know in the text it mentions stuff like canned goods and some like commercial production of things that uh, go through sterilization practices like autoclaving. Uh, and it truly depends on the product. If it's something that's got a lot of acid or a low amount of acid, different microbes have different potentials, and that requires more or less time, respectively. So we talked Louis Pasteur before. Pasteurization processes aren't completely killing everything. Typically, they're not. Uh, and what they're truly doing is bringing down the level of true pathogens uh, so that this product is safe. And I mentioned before with pasteurization, it was initially developed for alcoholic beverages, beer and wine, that would potentially get fouled with bacteria, and running it through a pasteurization process ensured that those microbes that potentially had got in there weren't able to cause problems. And when we uh, recognized the utility of that, we thought, could we not use that for other things? Like, how about milk? Uh, there are all sorts of organisms that we can see in milk, and definitely potentials for like raw milk, where these organisms still have the likelihood of being there. Um, I know they sell raw milk in different stores. You just hope that there's been a high level of uh, sanitization as they are dealing with the milking of those cows before it goes into your belly. Hard to say. So we see a handful here: Campylobacter, Coxiella, Listeria, E. coli, the bad one, Mycobacterium tuberculosis, paratuberculosis, Salmonella, and Yersinia intercolitica. All of these are potential pathogens that we could see in the cow. Ultimately, lots of it in the feces. Uh, so, two different ways for pasteurizing. The HTST, this pasteurization process is probably the most common. And if you go to the grocery store, most of the milk has likely been pasteurized through this process. So the meat, not the meat, the milk gets heated to 72 degrees Celsius. So basically brought up to a steam, not to a boil. It's brought to a steam and it's held at 72 degrees for 15 seconds. Then it gets bottled and refrigerated. Refrigerated, the key. This stuff has to be refrigerated or you're still going to see spoilage. We haven't got rid of all of the microbes, right? But these can't tolerate these temperatures and we've got rid of them, right? The ultra pasteurization, this goes at a higher temperature, 138 degrees Celsius, but shorter time. 2 or a few more seconds, it's held and then sealed and stored. And in this case, doesn't have to be refrigerated. All right. And you can definitely see some of these products at the supermarket on the shelf, not in the refrigerator section. And they are stable for a few months without refrigeration. Once you open that milk, it needs to be refrigerated, but it doesn't need it until that's occurred. There's definitely some differences with this as well. I am not a consumer of milk, but I know there are some differences. Uh, the taste of the pasteurized milk over here compared to this is slightly different. This temperature at 138 kind of gives it a different sort of taste as some of the sugars get acted upon within that, giving it, again, slightly different flavor. Raw milk, so I have been told, tastes different than all of these. All right. Uh, any raw milk consumers? Looking up to my participants. It's looking silent. I'm guessing no. All right. So, with that pasteurization, again, these are the ideas to get potential pathogens, to get their levels reduced so that those products are safe, right? Um, I'm sure you have all smelled some milk that's starting to sour a little bit as it was in the fridge a little too long. Um, Microbes are still present. That sets the stage for some of those issues. Not to mention the fact that you're opening and pouring it on your cereal and whatever, the potential for organisms to get in there just through that process uh, is also there. That's why you ought to put your milk back in the fridge after you ate your cereal. So we think heat as potentials for dealing with microbes, bringing them down to safe levels or potentially annihilating them. But there's other opportunities that we might think about, like chilling or cooling or freezing. Oftentimes we think about refrigerating uh, food to keep it safe, to ensure that potential microbes aren't growing. That's usually uh, a good practice, but there are some situations where some bacteria can still grow at refrigerator temperatures. Listeria is a good example of that. Other opportunities we can see here with this deep freeze. This looks like a freezer that you might have at your house or your parents' house, where they store, a side of beef or something like that. This is a bit different. It looks similar, but these freezers are ultra low freezers. We have numerous freezers like this in the biology department. Some still kind of deep freeze like, but most of them now are uprights and they look like the incubators in the lab, but freezers. Uh, they keep temperatures at negative 70 degrees Celsius. Some of them I have noticed are even a little colder than that, negative 85, uh, ultra low. And you see this guy opening the freezer up. He's wearing thick gloves. The freezer has three different areas. Each of these has like a styrofoam top on there. And as you open this door, you quickly pull this off, grab your sample, put the lid back on there, and close the lid up. It allows this other bit over here to stay cooler, so it's not just all open and exposed. And you want to be very quick getting in and out of there uh, to ensure that these samples all stay at that temperature. These typically contain all sorts of samples that we think about for preservation. Our bacterial cultures get preserved in these freezers for the ability to go back and keep those strains propagating. We freeze some of them away. Numerous researchers have all sorts of different specimens that they freeze in these to ensure that they're preserved and you can pull them out and continue to work with them. You can't just take the bacteria and put it into the freezer. It would ultimately cause that bacteria to die when it gets to that freezing temperature. We grow the bacteria up and we add a small amount of it in that broth culture. We add a small amount of it to some glycerol. Glycerol is super viscous, kind of syrup-like, and the glycerol provides almost like antifreeze, provides an environment to those bacteria that prevents those cells from exploding with that freezing temperature, and it allows them to stay viable. You can take that sample with your loop, grab a little out of it, pop it back into the freezer, and it continues to stay good. Um, Next to that in figure B, we see all sorts of steamy stuff. This looks like some sort of mad scientist lab. Liquid nitrogen is another mechanism to preserve things, and this is something that they oftentimes describe as snap freezing. Uh, you've maybe had an opportunity to see liquid nitrogen in action. If you haven't, I recommend when the chemistry magic show happens, usually around Halloween, I believe, is when that magic show happens in the chemistry department. They do all sorts of cool stuff down there, and one of them is some liquid nitrogen demonstration showing all sorts of different things, like a banana dropped into liquid nitrogen, and then they pull it out and hit it with a hammer, and it shatters. All sorts of other things. I think it's pretty awesome. Liquid nitrogen, super cold, and whatever it is that goes in there, basically the second that it touches the liquid nitrogen, it freezes it. And tissue samples, things like that, are oftentimes preserved in a situation like that where it's snap frozen and then able to be popped into one of these ultra freezers after that. So uh, you do definitely need to use caution with when working with the liquid nitrogen. I know in the room with the freezers, there is a canister of liquid nitrogen. I have never dealt with any of that stuff. I have used a little of it, but never went and collected it. And there's a warning sign right by the liquid nitrogen that basically says, if you are coming in here and getting liquid nitrogen, remember the buddy system. You should have someone in there with you to ensure that it is safe. You want to ensure when you go in there that you leave the door to the freezer room open as you are collecting it. If you are filling your canister up, there is the potential, if some of this was leaking out or whatever, it basically makes the oxygen level deplete dramatically and you could suffocate. As soon as I read that warning label, I was like, all right, mental note. I don't want it. I'm not messing around with that. Uh, so pretty interesting. And I think that's probably a good stopping point. Do you guys have any questions before I shut it off? Okay, I'm gonna stop it. I hope y'all have a good day. We will do another Zoom on Friday. Have a good day. Thank you, Matthew.